Welcome back to G Fun Facts Online. Today, we are walking into the woods, but not to admire the view. We are entering the world of the moss tracker. When you look at a forest floor, you might see chaos, scattered leaves, mud, decaying logs. It looks like a mess. But to a forensic ecologist, that ground is screaming with information. It is a high-fidelity recording device. Every step a criminal takes, every branch they brush against, and every moment they stand still leaves a biological barcode on them. This is the science of forensic botany and palynology, the study of pollen and spores. It is a discipline that turns the silent witnesses of nature into the most damning evidence in a courtroom. To understand how a speck of dust can convict a murderer, we have to start with the biology of the invisible. The primary actors here are pollen grains and spores. You might think of pollen as just the yellow dust that ruins your car in the spring, but under a microscope, these grains are marvels of biological engineering. The outer wall of a pollen grain or a spore is made of a substance called sporopollenin. This is one of the most chemically inert and durable organic substances known to science. It is nature's ultimate armor. Sporopollenin resists strong acids, bases, high temperatures, and decay. While a human body might decompose and a fingerprint might wash away in the rain, pollen persists. Paleobotanists find pollen from the Jurassic period that looks as pristine as the day it was released. In a criminal context, this means evidence deposited on a jacket in 1990 is still there, chemically unchanged. 30 years later, it waits. But durability is only half the story. The other half is diversity. There are roughly 400,000 plant species in the world, and each one produces a unique pollen grain. Under a scanning electron microscope, they look like alien spacecraft. Some are spiked spheres, like the sunflower family. Others look like geometric lattices. Some, like pine pollen, have little air bladders that look like Mickey Mouse ears. A forensic palynologist can identify the plant from a single grain. However, the real power isn't in finding one grain. It is in finding the assemblage. Imagine a specific patch of woodland. It has an oak canopy, a hazel understory, a specific patch of stinging nettles, and a rare moss growing on a rotting log. The air and soil at that exact coordinate contain a specific cocktail of pollen and spores that reflects that exact community. 100 meters away, the mix changes. When a suspect walks through that scene, they become a pollinator. They pick up that specific cocktail on their clothes, in their hair, and even inside their nasal cavities. This acts as a forensic GPS. If we find a mix of 40% oak, 20% hazel, and 5% of a rare moss on a suspect's shoe, and that mix exists nowhere else in the region, the probability of them having been there is overwhelming. Now, let's talk about the title of today's episode, the moss tracker. While pollen blows in the wind, moss is different. Bryophytes, which include mosses and liverworts, are the heavyweights of forensic botany. They don't fly far. If you have fragments of a specific moss on your clothing, it implies direct contact. You didn't just walk near it, you touched it. Mosses are also incredibly resilient. A scrap of moss can dry out completely, looking like brown dust. But in the lab, when a scientist adds water, it can rehydrate and unfurl, revealing its distinct cellular structure. DNA can be extracted from these tiny fragments years later. Because mosses grow in very specific microhabitats, some only on limestone, some only on acidic bark, finding a specific moss assemblage is like finding a receipt with the GPS coordinates of the crime scene. How do scientists actually get this evidence? It is a meticulous process. It starts with collection. Investigators take samples from the crime scene, but they also take control samples from 5 meters away, 10 meters away, and 50 meters away. This establishes just how unique the crime scene is. They also take alibi samples. If a suspect says they were playing football at the local park, the scientist goes to that park and samples the grass. Then, they compare the suspect's shoes to both the crime scene and the park. Back in the lab, the evidence undergoes a process called acetolysis. This is pure chemistry. The samples are boiled in a mixture of sulfuric acid. This aggressive bath dissolves everything: cellulose, fats, proteins, bacteria, except the sporopollenin. The pollen is the only thing that survives. The scientist then spends hours at a microscope, counting thousands of grains to build a statistical profile. This science has solved some of the most notorious crimes in history. Let's look at the Soham murders in 2002. This case changed everything for forensic botany in the UK. When 2 young girls went missing, the school caretaker, Ian Huntley, became a suspect. But there was very little physical evidence linking him to the remote ditch where the bodies were eventually found. Enter Professor Patricia Wiltshire, the Sherlock Holmes of Spores. The bodies had been found in a ditch overgrown with stinging nettles. Now, nettles are common weeds. Finding nettle pollen on someone's car normally wouldn't prove much. But Wiltshire went deeper. She analyzed the pollen found on Huntley's car and shoes. She found high concentrations of nettle pollen, but crucially, she found clumps of immature pollen grains. This was the smoking gun. Nettle pollen is usually released into the wind as single, dry grains. Clumps of immature pollen only exist if the plant has been violently thrashed or trampled, forcing the pollen sacs to burst prematurely. The presence of these clumps on Huntley's items proved he hadn't just walked near nettles. He had driven through them and trampled them violently, exactly the action required to dump bodies in an overgrown ditch. Furthermore, the soil on his car contained a specific mix of brick dust, concrete, and moss spores that perfectly matched the track leading to the site. The plants destroyed his alibi. Another incredible case involves the cold case of Christopher Laverack. In 1976, a 9-year-old boy was murdered and found in a river, weighted down by a heavy ornamental brick. The case went cold for 30 years. When it was reopened, scientists analyzed the pollen on the boy's clothes. It revealed a garden assemblage, privet hedges, cypress trees, and ornamental plants. This didn't match the riverbank. It suggested he was killed in a domestic garden. The focus turned to the brick. Even after being submerged and stored for decades, the crevices of the brick held a secret. It contained the same garden pollen, plus spores from a fungus that grows on rotting timber, and pollen from a specific type of cedar. Investigators looked at the boy's uncle, who had died years prior. They examined the garden where the uncle lived back in 1976. Despite 30 years passing, the fingerprint of place remained. The garden had the privet hedge. It had the cedar tree. The pollen profile of that garden was a statistical match to the pollen on the murder weapon. The plants held the memory of the murder for a generation, allowing police to identify the killer posthumously. This technology isn't just for murder cases. It is used to track drug shipments. Cocaine and heroin are plant products that grow in specific regions. By analyzing the pollen trapped in a brick of cocaine, intelligence agencies can tell if it came from the Chapare region of Bolivia or a specific valley in Colombia. They can even trace the route the drugs took by the pollen picked up during transit. If a shipment contains pollen from a pine tree that only grows in Morocco, they know the smuggling route. It is also used in investigating war crimes. In the aftermath of the Bosnian War, perpetrators often moved bodies from mass graves to secondary locations to hide the evidence of genocide. Forensic palynologists found that the bodies in the new graves were coated in pollen and soil types that didn't match the new burial site. Instead, the pollen matched the geology and flora of the original execution sites. This provided scientific proof of the cover-up and was used as evidence in international war crimes tribunals. So, what is the future of the moss tracker? It is getting high-tech. We are moving from microscopes to DNA sequencers. This is called eDNA or metabarcoding. Instead of visually counting grains, scientists can run a soil sample through a sequencer that reads the DNA of every plant fragment, bacteria, and fungus in the mix. This generates a massive, digital biological fingerprint with unparalleled resolution. We are also seeing the rise of AI. Counting pollen is slow and hard on the eyes. New AI systems are being trained to recognize pollen grains instantly, which will allow for global databases of pollen maps. Imagine a world where a detective can upload a pollen sample and a computer instantly pinpoints the location on a map, like a reverse image search for dust. The concept of forensic ecology reminds us of a profound truth. We are not separate from nature. We move through it, and it moves with us. Locard's exchange principle states that every contact leaves a trace. Plants are the ultimate adherents to this principle. They are designed to disperse. They want to travel. When a criminal moves through a forest, the forest is biologically engineered to tag them. Burrs stick to wool. Sticky sap adheres to denim. Microscopic hooks on spores latch onto synthetic fibers. The criminal isn't just leaving traces, they are being colonized by the crime scene. They might wear gloves to hide fingerprints. They might bleach a room to destroy human DNA. But they cannot stop the wind from blowing. They cannot stop the moss from shedding its spores. As long as they step on the earth will remember them. That is the power of the moss tracker. Thanks for listening to G Fun Facts Online.